I'm Amy Donahue. I'm the user education and one of the reference librarians at the MCW Libraries. And today we're going to be talking a little bit about NCBI's molecular databases, just kind of a really basic brief introduction to a few of them. I'm first going to go over what NCBI is and what it does just a little bit. Then I'll introduce the specific databases that we're going to look at. So those are gene, protein, and nucleotide today. Towards the end, I will do a quick demonstration of the NCBI global query search as well. And then I have some actual hands-on practice exercises at the end for you to play with. So yeah, by the end of the class, hopefully you'll understand the mission of NCBI. You'll be able to do some basic searching within gene, protein, and nucleotide, as well as kind of understand a little bit and be familiar with what those records look like. You'll have an understanding of how the NCBI databases are connected. And then finally, you'll know where to find help and support to continue using all of these great tools. So what is NCBI? It stands for the National Center of Biotechnology Information, and it's a division of the National Library of Medicine, which actually itself is one of the 27 National Institutes of Health. So ultimately, NCBI is part of NIH. And it is a national resource for molecular biology information. There's a lot of text on this slide, but basically what NCBI does is develops, distributes, supports, and coordinates access to a variety of databases and software for the scientific and medical communities, with the ultimate purpose being to advance science and health by providing access to biomedical and genetic and genomic information. So NCBI actually has developed well over 50 databases. There are a lot of them, and other resources. And those include PubMed and PubMed Central, as well as Gene, Protein, PubChem, GenBank, DBSNP, Taxonomy. These 50 databases span chemicals, genetic expression, clinical genetics, protein structures, and the biomedical literature. And I will note that there is overlap in the content. So information is reused in the different databases in different ways for different audiences, but some of that same content is there. And furthermore, there is definite linking between the different databases. So if you're in one database, you can jump easily into another, and I'm going to go ahead and demonstrate that in just a few minutes. And I'll also note that the NCBI databases are free and public. Anyone can use them internationally. Some data in some of the databases is controlled access. You do have to register for an account for research participant privacy. So the major takeaways from this slide, which again has a lot of words, just wanted to highlight that there are a lot of databases, and those databases are all interlinked. So these are the three databases we're going to focus on today. The first one is Gene, which is a searchable database of genes focusing on genomes that have been completely sequenced and that have an active research community to contribute the gene-specific information. The records provide information on individual genes. So we'll take a look at those records, but that's what the type of information you'll be looking for when you go into the gene database. Protein is a collection of protein sequences. So I just wanted to highlight that word sequences. If you're looking for a sequence, the protein database is where you're going to want to go. And these sequences come from a number of different places. And then nucleotide is very similar to protein, except it's a collection of nucleotide sequences. And again, these come from several different sources. So I'm actually just going to walk through a demonstration that goes through all three of these databases, kind of centered around the cystic fibrosis CFTR gene. So I'm going to go ahead and just click the link to go into NCBI and go to their homepage. I'm going to try to zoom in just a little bit. But I just want to point out a couple things on the homepage. They do have their popular resources over here. There's a lot of information. NCBI does have a blog and various announcements, and you can get on their mailing list. I'm on it. It's really very useful. But their resources are kind of grouped over here on the left-hand side, so you can drill down into the area that you're particularly interested in. Or you can just search right away from the top. Select the database you want, and you're good to go. Just as an example, I'm going to go ahead and go into the genes and expression area just to show you what these look like. There's my list of databases in this particular group, and I'm going to click into gene. A couple things that I'm going to point out on this page as well before I do my actual search. All of the NCBI databases tend to have this banner right up at the top where it tells a little bit about the database, and then right underneath that you'll see some help files. So this is a good place to go to get support right away. There'll be an FAQ, lots of times there's a fact sheet, so this is across all of the NCBI databases. And then also underneath here you'll often find related tools or resources for that particular area. But on the gene database specifically, underneath that, they do have these representative queries that I just wanted to point out. If you're familiar with searching in PubMed at all, you can search by fields in PubMed by like author or title, kind of those basic citation sections. But you can do actually the same thing in genes and in the gene database, and these representative queries kind of show how you can do that. So you can just do a free text search. You can also search for a particular chromosome. So I just wanted to point that out, that that is an option. And I will demonstrate one way to take advantage of those in a few minutes. But first I'm going to start with just a basic search for my CFTR gene. Just typing it into that text box and doing my search. So here's my results. They do give you this nice table. You can change the display to be a summary text format, but the default is this nice table that shows you the description, the name, the location, lots of useful information. On the left-hand side, there are ways to drill down your search results, just like in almost all of the NCBI databases. So a lot of these things will seem familiar as you use all of the different tools, but you do have to be familiar with what the filters are in a particular database. So for instance, for this particular search, I am only interested in the genomic sequences, not so much the mitochondrial. So I can use that to narrow down. I think it might be a browser issue too. If you click things and things don't seem to be working, feel free to give the libraries a call and we can help you troubleshoot. What would normally happen when I click this is that then it would jump from being 2,900 results to being about 2,700 or so. It gets rid of those ones that I don't need. And also over here on the right-hand side, I just want to point out the organism so that you can drill down to the particular organism and you're interested in really quickly as well. But in this case, the gene I am looking for really is right at the top, so I don't need to worry too much about trying to dig around for things that are particularly relevant. So I'm just going to go ahead and click into the record. And again, just going to go over what you're seeing here on the screen. So at the very top of all of the gene records is this nice little summary that has the official symbol, the official full name, and a lot of other information. And anytime you see this blue text with the underlining, that's something that you can click on and either link out to another database or another original source of the data, things like that. For instance, if I clicked on Homo sapiens, it takes me into the taxonomy database. Over on the right-hand side, there is a table of contents, so you can jump down into the record to wherever it is that you need to be. I'm going to actually scroll through the record, but just so you're aware that that is something that can help save you time, especially if the record is really long. You can jump right down into what you're looking for. Also on this right-hand side is the related information, and I'll probably come back to this a few times, but this is where you'll be able to jump between the databases all on the same topic. So all of these databases, if I clicked on it, it would run the same search on CFTR, and particularly related to this gene record. So it would link, there is a connection between this gene record and the results that would come from any of these related information databases. So like I said, I'll probably repeat that, but going back to the main record side and scrolling down a little bit farther, I just want to point out a few things in the sequence viewer section. This is kind of fun. I like to play with it. There's a lot going on here that I don't necessarily understand at all, but I do know a couple of tips and tricks that I'll just point out here. So I'm going to first change my sequence to the RefSeq sequence, which is, RefSeqs are a set of reference sequences that are curated standard sequences for a gene. So not every single gene record will have one of these, but when they do, you'll see some extra information there. So when I pull up the RefSeq sequence, I'm now also going to click on the gene line itself and just expand that so that now I can also see the mRNA and the actual protein along the viewer as well. And if I click it again, it'll go back to the one line. Just some handy navigation stuff right here. I'm also going to point out a way to zoom in. So let's say I'm interested in kind of this exon 12 section. I can actually just left click at my starting point and then scroll through the base pairs until I reach the end of the area that I want, let go, hover over it, and then I can zoom on that particular range. So now I'm in this area. I'll point out here the SNP histogram that's underneath the sequence. So anywhere where it's darker, there's going to be a lot more SNPs. So that's what that's representing. And I'm going to zoom in a little bit farther even, and this time I'm going to double click on the exon that I'm interested in. And that will zoom it in right to that area. So now I can really see the area where my SNPs, there are a lot of SNPs. And I can also see what's going on in this clinical channel. Anything that's green is an allele that has been found to be benign. It's unknown, but it's almost definitely benign. The purple ones are actually pathogenic alleles. And again, anything that's underlined in blue is something that you can click out to. So you can click between those variations and get to that information quickly as well. The light purple means it's unknown, but possibly pathogenic or likely pathogenic. So that's what those colors mean. There is help for all of these areas in the record. That's a good question. Where can you find the color codes? These question marks for each little area in the gene record, if you click on those, will take you into information for that particular area. I don't remember exactly where the color explanation is, but it's somewhere in this help file. So, but purple is that clinically relevant allele or variant. This is a different exon. I'm in 12 now, but the last thing I wanted to point out just is the cited variant. So down below the clinical channel, you can actually see the variants that have PubMed records associated with them. So you can go back and get that actual original research as well really quickly. So I'm going to continue scrolling down now. There's a lot more you can do with this viewer. You can configure it in different ways. So if you ever want more help exploring any of that, we'd be happy to do it as well and help you figure out what's going on and to do the things you need to do with it. But below it is the bibliography section, and this is where you can actually jump to all related articles in PubMed. They'll show 5 to 10 that are on this kind of short list, but there's also a link to all of the citations related to this particular gene record. And below that, also part of the bibliography, is this gene riffs or references into functions. And they have a nice little explanation of what those actually are. But they are concise phrases describing a function or functions of a gene with the PubMed citation supporting that assertion. So these are actually phrases that come from a PubMed record that explains what this gene does. And if you click on it, it'll take you back into that PubMed record. But the phrases themselves are here, which is a nice little summary of that gene's function. And continuing down, in the phenotype section, I did just want to point out that cystic fibrosis is just one of the phenotypes associated with this gene. And again, we can jump back and forth into other databases to learn more about these particular phenotypes, for instance. So there's more information down below. Those are kind of the big areas that I wanted to point out. I'm going to kind of jump down to, there's a lot more here, the general protein information, because here's where I'm going to jump into the protein database. You'll see our sequences. The RefSeq is at the top, and there's other sequences here as well. And again, you can link to anything. But what I want to point out is the NP protein number. That's what I'm going to actually grab. I could just click on it down here to get into the protein record, and I can also click on the protein link in that related information up on the right-hand side. I'm going to demonstrate just really quickly a third way of doing the searching. Go back up to the top. And I'm just going to search the protein database from my list up here using that protein number. I could also search it for CFTR again, but I want this particular protein, and this is actually going to take me right into the protein record. So protein records are a lot more simple compared to the gene records. They're all text-based, but they do still have the linking, so I can jump back and forth, PubMed numbers, for instance, into PubMed. This is a RefSeq, so again, this record is annotated. It's got a lot of information associated with it, including this comment from the annotators. But as we scroll below that, then we start to get to all the protein information, its location, the sites along the protein. And I'm going to keep scrolling through all of those, but there is information in each of these areas, like where it came from, some notes for all of those. But what I'm getting down to is at the very bottom is our actual sequence. So there's the sequence for our protein. And then you could copy that as you needed to run other searches as well. And again, now that I've scrolled all the way through, I'm going to scroll back up and just point out again, here's our related information. So even from the protein database, we can jump back into gene. We can look at a number of other systems. And actually, I'm going to point out one that wasn't on our original list, and that's just the related structures, just because I think it's cool. But in addition to all this text data, of course, we can get the actual structure data related to our particular protein as well. So these proteins can be viewed in the CN3D viewer from NCBI, which is a free tool. It lets you manipulate the proteins and change how you're viewing it. You can do the rods. You can do, I forget all the different names for the different types of views, but it can really change your understanding of how the protein structure is working. So you can link right into those as well. So that's gene and protein. And now if I want DNA sequence data, so I'm actually going to jump into nucleotide and end with our third database here. So the nucleotide records, if I click on it, it's going to find all the records that are related to that particular protein. So there's more than one, but I'm going to scan through my list, and the one I want is, the Homo sapiens CFTR RefSeq gene on chromosome 7. You can also take a look at the accession numbers. They do give you a little bit of information. I don't remember offhand every single one of them, but the NG number is a gene record. So it's related to that gene that we originally started with. So if I go into that one, you'll see that the nucleotide record is very similar to the protein record. Again, the summary information up at the top, and then PubMed references, the comment for the RefSeq record. And here you'll see the exons, the coding sequences. There's a long amount of data here, and then the translation from that coding sequence. And to save a little bit of time, if you actually click on the coding sequence space, it'll jump you down lower in the record to the sequence itself, and even that particular coding sequence section. So you can actually click there to jump around in the record as well. So I'm going to click the little X to remove that, and as I scroll back up, you can see this is a very long sequence, but that data is just at the bottom of this particular nucleotide record. There it is. That's, the really quick introduction to those three databases and kind of interlinking between them. Just jumping back to my presentation really quickly. Now I'm just going to go briefly over that kind of global cross-database search that I mentioned at the beginning. So this search just allows you to search across all of these databases at once. And I'm going to go ahead and demo that in just a second. But just a couple quick tips. You can actually do it from anywhere in NCBI. Just from that drop-down menu, you change that to all databases, and I'll point that out. But do keep your searches simple. You're searching across databases, and these databases have different fields. So PubMed has titles, and Gene Records has symbols and things like that. So you want to make sure you're maybe just searching one phenotype or the name of a gene or something like that. But go ahead and try it, whatever you're looking for, and see what results you get. You can use Boolean searching, so you have to use the all caps and or not to search in the GQuery box. And then I just wanted to briefly mention that you can save individual searches from any of the databases on the GQuery results page, and I'll point that out. You can't save the entire thing, but once you're in any single one of them, you can save that particular search. So going back into NCBI, I'm going to go ahead and use that drop-down box and do all databases. And if you just do a blank search in any of these, it'll take you to that particular screen's homepage. So if I did it for protein, it would have taken me to the protein homepage. But just this is the GQuery interface. I'm just going to do my CFTR example again. And then it just quickly searches across all of them, and I can see how many records are in all of these different places. And I can see they are grouped, so here's my clinically relevant databases that I didn't go over today, but here's gene under genes, 2,911 results, which is what we got when we searched gene alone for CFTR. So I'm going to go ahead and just jump into the resource that I wanted and point out that Save Search feature. So once you've gotten into the database that you want, you can just click the Save Search, and again, this is for any of them. You'll be prompted to log in or create an account with my NCBI, but it's a really simple process. You just need to provide a username, password, and email, and then you'll be all set to save your searches. You can rerun them as new information is available. You'll have an easy place to keep them for future reference and all of that good stuff. Just going to go back to the GQuery page. Any questions about that kind of federated search? Yeah?
So is the best strategy then to start with GQuery so that you could kind of see where the most information is or kind of where the distribution of data might be? Or is it an it depends question?
It's an it depends question. For us, for librarians who might need some background or for just a really general question, this probably would be a good place to start to see where that information is. And it's also nice to see, okay, so I have 31 interaction studies. I know exactly what's in dbGaP just from reading the little information here. But if you know what you want, if you know you're looking for sequence data, then you could do a quick search in GQuery to see if there are things in both protein and nucleotide, for instance. So it depends. Some people know exactly what they're looking for and know which database to go into. But these can also be helpful if you're not sure what other databases might have information. So if you already are familiar with the protein record and want to see what might be in some of these clinical results, this might be a good way to go. And it's also good for things when you're not finding any information. You can do a quick search in GQuery to see where else there might be something. So the final slide in my presentation is just the help and support. So I pointed out those using resources. That's what they always call them, using gene or using protein, that's underneath that banner on the home page. So that's where you'll find resources for specific databases. So it's kind of on the bottom left. One other little help section that I did not point out yet is this how-to, which is always at the top of any NCBI resource. This blue bar will always be there. And they actually provide very clear step-by-step how-to tutorials, basically. So if there was some specific thing that I wanted to do within the genes and expression area, I can actually click that how-to and walk through those steps. So those are available as well. So those are the NCBI resources, and they do have things like YouTube videos that will be linked to depending on the resource and all of that good stuff too. Things to read, things to watch. But then the MCW libraries can also help support some of this. We can offer additional training. This was really brief, quick introduction. I've had some training in some of these databases, and we are happy to spend the time to become better experts to help you so that you don't need to spend lots and lots of time trying to figure this stuff out. We can also provide reference services, so not just the PubMed literature searching, but if you have some specific question about a gene or about biomolecular information and you can't find the answer, comparing sequences, things like that, we can help with that for sure. So feel free to think of us. And with that, the actual presentation part of this class is over, but I do have some practice questions for you if you want to stick around. So we'll start with the how many human genes have been linked to Alzheimer's disease. And here's where I'm going to show you that advanced search in gene that I did not do before. So I'm going to go into my gene database just do a blank search to get to the home page. And then under the search bar, and this is again true across almost all of them, if not all of them, you'll see this advanced option. So for PubMed, for instance, it's also definitely true. And if you click on that, you'll be able to build a search using the available fields. So what I'm going to do, you can scroll through and find whatever it is that is the closest to what you're looking for. And then you don't have to worry about getting the field names correct and all of that. So here's my disease phenotype. And I'm just going to start to type in Alzheimer's. And after I get the first few letters, I'm just going to show the index list. And this shows me all of the words that have been indexed with this particular string of letters. So I can find all of the records that have Alzheimer somewhere in that particular field. And I can actually select multiple too. These are all just repeats, so Alzheimer will include all of these Alzheimer disease, 5, 6, 7. But if there are multiple that you want to select, you can actually use the control button to select individual ones. So I'm holding down control and clicking those. Or if I wanted to select a big group, I can hold down shift and just click through so that I've got all of those at once. I'm going to stick with just that basic one. And I'm also going to limit, in this case, I know that my organism that I'm interested in is definitely humans. So I'm going to do the same thing. Just to confirm, it is human, that is correct. And then just click search. So here is a nice little table of my 45 genes that are associated somehow with the phenotype Alzheimer's. And here's the display settings. Somebody asked about changing the sorted by. So by default, NCBI is switching over to this relevancy ranking. But you can also, depending on the database, do it by different things. So in Gene, you can do it alphabetically, for instance, or by the chromosome location. In PubMed, you'd be able to do it by the publication date or the added date, which can be different. Things can be added to PubMed before they're published, so those are the two different dates. But that's how you change that. And you can also do all sorts of things. Here in Gene, you can change the table to a summary text. You can change the number of items as well. So you have that power. So that's the advanced search. Just to quickly demonstrate, I'm going to go ahead and just put in, now that I have that correct field code, I know how to spell asthma. So I'll just change that just to show that it's kind of interesting that asthma, kind of perhaps to be expected, has a lot more genes associated with that particular disease. So that's the way I would have done it. You can just type in asthma and do the search and look at all your genes. But just know that it might be in some other field when you're doing it that way. In this case, I know exactly that the asthma is in the disease phenotype field. But if I'm interested in anything that might have any relation to asthma, then I might want to go ahead and search across all of them. So it's just kind of something to be aware of. For my next question, right, my T-Box 1. So this was a story that was just on the front page of the MCW website, and I thought it was interesting, with a woman with 22q deletion syndrome. And what was interesting was how long it took them to diagnose it. So in this case, I am just going to go ahead and do my regular search, because for this one, it's more just the playing around in the record that I wanted you to do. But my house mouse does come up first. I want my human. I'll go ahead and jump into the one right there. But in this summary, it has this information about what the T-Box genes do. And they are involved in the regulation of developmental processes. So it's very early on, and it just leads to all kinds of things from cardiovascular to neurological, and there's all these different symptoms. And it's a domain that's conserved along almost all animals, actually. So almost every animal organism has the T-Box sequence. And then from here, you can go ahead and jump down to that protein and nucleotide information. So I'm going to go ahead and do it quickly since I know what I want. I want the protein information. I jump down there. In this case, there's not a specific NP name associated with it in this area, but there is in the sequence. So I can jump right into the one of the proteins. I think there's actually a few isoforms, but I can just jump into the isoform B as the example right now. And then from here, If I scroll down, protein, actually a few of these have a lot of information on the right-hand side. It kind of depends on the record and what you're looking at, but I'm looking for my related information box, and then I can jump into nucleotide to get to that sequence information. And again, there's more than one. There's no NG records. I'm going to go ahead and jump into the NM, which I believe is associated with the mRNA. Yep, mRNA right there. And then I can look at my sequence information and find the line that starts with 601 to get that particular piece. Oh yeah, and I did give the hint to use the little ref-seq sequence. Right, I'm going to point that out really quickly again. If I go back to the gene record itself, and one other way to get that NP number really quickly, if I get the ref-seq and get that one protein, oops. So there's my NP number associated with it. So I could just get that number right there as well. So that's what I was hinting at with the ref-seq information as well. So this is just basically kind of a repeat of that first one. Did anyone look up anything interesting that they wanted to share? If you actually try the advanced search with your disease of interest. So my little example, can bring that up. And the number next to it is the number of results that are indexed with that particular one. So I know I only have 3, so that makes it easy. So my TP53 gene, I can click into there, but there are these 3 genes associated with this particular phenotype. But in terms of the pathogenic alleles then, where I would find that is actually on that reference viewer. Again, going to the reference gene so that I can see that clinical channel. So on this particular gene for Li-Fraum-Any syndrome, there's 1, 4, 6, 13, 18, and these have a lot. So you just kind of add those up and just recognize that there's over 20, probably over 30 different allele places that are pathogenic. And then the PubMed search. So I can actually do a PubMed search right away from my drop-down menu, so I'm going to go ahead and do that. I'm just going to pull up PubMed from my list, do a quick search on the same phenotype, and here are my 1,029 articles that have been mapped. PubMed does fancy things with mapping my words to mesh headings as well as doing the keyword searching, so it's found all of those articles for me. But the way to get from a PubMed search results page into the gene information, so this is from the results page, scrolling down the right-hand side, this time there's not a related information tab or area, but I can actually find related data by clicking on the database and then clicking on gene, for instance, and then it will pull up gene records that cite the current articles for me. So even from a PubMed search, which is where I might start if it was something that I didn't know very much about, I can then get into the gene records for that particular thing. And you can do the same thing, usually within any individual article. Okay, actually within the individual articles, they do have the related information. And if there is a gene record, this is where those numbers come into play. A gene record associated with that PMID, it would also show up here. There'd be a link to gene. But right now, there isn't anything actually making that linkage. So in some ways, doing it from the results page is actually can be a little bit more fruitful. But the other thing that happens if you're actually searching for a gene, and I think some of you stumbled upon this, is that PubMed will pop up this helpful little box right up at the top that will take you into the record in the gene database. And then you can also find it for these common organisms, as well as take you specifically to the articles that are about the gene function. So that's a nice little subset out of these 18,000 articles that might be more useful. As well as if there is genetic testing available, this is kind of an interesting little thing. Another database does have the contact information and the places that will do testing for a particular gene. So that's there as well. So that was the bonus PubMed question. I think that wraps everything up.